In this video, we're going to talk about how different types of epithelia can be classified. So I'm including this table here at the outset, which you can refer back to later when you're reviewing or putting together your study guide. Now, generally speaking, epithelia can be classified anatomically or physiologically, but these two classification systems are overlapping. And if you take a closer look at this table here, you'll probably see some examples of that. We're going to talk first about functional classifications of epithelia, starting with exchange epithelia. These are often located at the interface between two different compartments, and they allow molecules to move between two adjacent cells paracellularly. And by doing that, they allow for rapid, non-selective transport of substances between two compartments. Similar to exchange epithelia, we also have transporting epithelia. These also allow solutes to be exchanged between two compartments, but they have much greater selectivity. And this is because transporting epithelia are usually held together very, very tightly, which forces molecules to travel transcellularly through the cell. And that means that the molecules have to cross membranes, and we know that membranes are selectively permeable. Many epithelia can also possess cilia on one of their membrane faces. This is especially common in places like the respiratory system and the reproductive system. Cilia that are on adjacent cells typically beat in the same direction, and that creates laminar flow through the extracellular environment. That's really important for moving solutes. If you look closely at the video that I'm playing here, you can actually see the coordinated beating of cilia, and you can see some solute particles moving through from left to right across the field of view. Cilia can also play an important role during development because their coordinated beating in the same direction helps developing cells to orient themselves in 3 dimensions, similar to how you could use an air current produced by a fan to guide your movements if you were blindfolded. This is actually one of the major reasons that certain types of ciliary dyskinesia, which are disorders characterized by abnormal ciliary movements, often impact the growth of tissues. One example is Cartagener syndrome. Cartagener syndrome is characterized by respiratory problems similar to cystic fibrosis, but a large percentage of people with Cartagener syndrome also have situs inversus, which is when the internal symmetry of your organs is reversed. So, for example, if you take a look at this picture, you might find your liver in your upper left quadrant instead of your upper right quadrant. Now, the reversed internal symmetry actually doesn't produce any major problems in most people. So one of the interesting things here is that it's possible to have this condition without even being aware of it. Oftentimes people will become aware of it when they go in to seek treatment for some other condition like appendicitis, and the surgeon's like, something doesn't look quite right here. Epithelial cells can also be responsible for secretion inside the body, and that's because clusters of epithelial cells can organize themselves into glands. Glands can be further characterized based upon where they release their secretions into. So many epithelial glands release their secretions into the bloodstream, and these glands are known as endocrine glands. We're going to be talking a lot more about endocrine glands towards the end of our course. They can also organize into exocrine glands. And exocrine glands, on the other hand, don't release into the bloodstream. They release secretions into ducts, which will carry them to some kind of external environment, like, for example, the surface of your skin or the digestive tract. We're going to see a lot more examples of exocrine glands in the second half of the course later on. Now, because there are so many different types of exocrine glands and endocrine glands, we can further classify them anatomically based upon their 3-dimensional structure. Here we're looking specifically at exocrine glands. And so we could classify an exocrine gland as simple if it contains a single duct, or compound if it contains multiple ducts. They could also be classified based upon the overall arrangement of cells in the gland, with a tubular exocrine gland taking on a tube-like shape, whereas an alveolar exocrine gland takes on a more bulbous or mushroom-like shape. So in this diagram, the secretory cells of the gland are in dark purple, and those are referred to as acinar cells, whereas the cells that make up the ducts are in pink, and those are nicely named ductal cells. We can also classify exocrine glands functionally based upon the mechanism of secretion. Most of the glands that we talk about in this course are merocrine glands, which means that they package their secretions into membrane-bound vesicles, which can then be exocytosed from the cell in response to some kind of stimulus, usually a hormone or a neurotransmitter. These are commonly found in the digestive system, and that's where we'll talk about them the most. Apocrine glands, on the other hand, move their secretions towards one membrane face, because remember, these are polarized cells, and that membrane will then engulf the secretions and pinch off into the external environment. And when it does that, it carries the secretions away with it. Lastly, the holocrine glands release their secretions by rupturing cells. So this typically happens in glands that have very lipophilic secretions that would interact with the plasma membrane. If they interact with the plasma membrane, it would make it difficult for them to be released from the cell. They would get stuck in the membrane.